Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 5 for January 2023. Let us begin with the first question. Question 1 says, the diagram shows an elbow joint. So we have the humerus bone, which is that, and we have the ligament, the radius, as well as the ulna. So they say which structure contains actin and myosin. Actin and myosin are contained in a muscle, so the answer here should be a C. Moving on. Here they say, what is the function of the part of the joint labeled W? So if I go back here, W is a muscle, and because this is a flexor, this is going to be an extensor. So my answer here is it acts as an extensor. In part B, explain the role of the ligament labeled on the diagram. When we go back to the diagram, this is the ligament they're talking about, and as you can see, it's joining the humerus bone to the radius bone. So here I said it joins the bone in the upper arm, which is the humerus, to the bone in the lower arm, which is the radius, so that their position in the arm is maintained. Then down here they say suggest one possible cause of damage to this ligament. This ligament could tear during sports or intense activity, or it could be damaged during bone dislocation. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Different techniques can be used in the investigation of the activity of the brain. Which type of scan can be used to study changes in brain function as they happen? This is studying them during activity. That is usually functional magnetic resonance imaging, which is fMRI, so the answer here should be a C. In part B, describe the advantages and disadvantages of using positron emission tomography in magnetic resonance imaging to investigate brain function. So here we're going to talk about the advantages and disadvantages of each of the imaging technique. So I began with PET, which is positron emission tomography. PET can be used to detect cancer growth by studying which regions of the brain are metabolizing. However, since PET uses radioactive tracers, there is exposure to radiation. Also, images are 2D and have low resolution, so this is a disadvantage as well as that. When we go to MRI, the advantages are it gives 3D images with great resolution. Also, there is no ionization radiation. However, MRI is expensive. It confines in enclosed spaces, so if somebody has a phobia about that, then they'll be affected. Also, it's noisy and can't work for people with implanted metallic body parts because, of course, the magnet will rip the metal out of the person's body. Moving on. In part C, the diagram shows a section through the human brain. So we see the cerebral hemisphere, we see the hypothalamus, the pituitary gland, as well as the cerebellum. Here they say complete the table to show one function of each part of the brain. The cerebellum is the region where balance and maintenance of body posture is obtained. The cerebral hemispheres is a region for intelligence, memory, critical thinking, emotions, and so on. And then the hypothalamus is for osmoregulation, thermoregulation, or you could say for homeostasis. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Nervous communication involves neurons and changes in ion concentration. In part A, which description of the movement of ions will produce an action potential in an axon? An action potential is produced when depolarization occurs due to the movement of sodium ions into the axon, so this is going to be my answer. Then next they say, which row describes the events that occur during repolarization of a neuron? Remember, we have something like this. It goes up and then down. So this is during the repolarization state. So they're asking which row describes the events that occur during repolarization. In repolarization, sodium ion channels are going to be closed and the membrane potential decreases. You can see, of course, this is going down. So no more sodium ions are coming into the membrane to prevent the potential from increasing. Moving on. In part B, describe the structure of a spinal reflex arc. The spinal reflex arc begins with the receptor cells and ends with the motor neuron synapsing with the effector. So I said receptor cell synapse with a sensory neuron, and then the sensory neuron will synapse with a relay neuron. The relay neuron synapses with the motor neuron, and the motor neuron synapses with the effector for a response to be carried out. In part C, explain how the nerve impulse is transmitted across the synapse. Inside the synaptic cleft, we have calcium ions. So this begins with the voltage-gated calcium ion channels opening and calcium ions diffusing from the synapse to the presynaptic neuron. Then the calcium ions will bind to the vesicle and cause them to fuse with the presynaptic membrane. Here, neurotransmitters are going to be released into the synaptic cleft, and then the neurotransmitters are going to diffuse across the synapse to the postsynaptic membrane. Then the sodium ion channels will open, leading to depolarization or generation of an action potential. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Cancers can develop as a result of mutations or epigenetic modifications. State what is meant by the term mutation. A mutation is a random change to the base sequence of DNA. Next, they say, name the type of nuclear division taking place as a tumor develops. This is going to be mitosis because cells produced are going to be identical in order to produce the tumor tissues. In part B, how many of the following statements about epigenetic modification are correct? It results in a change in the base sequence of a gene. It does not necessarily change it. It can involve histone modification. That is correct. It can result in an altered phenotype. That is correct, because different proteins can be formed due to switching genes on and off. It can involve DNA methylation. That is correct. And again, to clarify, epigenetic modification just switches off or switches on genes to control which are going to be transcribed or not, and it has no hand in changing the sequence of the gene. So here my answer is C. Only 3 statements are correct. Moving on. In part C, estrogen stimulates some types of breast cancer to proliferate. Estrogen is a steroid hormone found in mammals. Estrogen affects up to 100 different genes. Explain how estrogen may result in the proliferation of some breast cancer cells. Because estrogen is a steroid hormone, it's going to enter inside the cell and act as a transcription factor. So I said estrogen enters the cell through the cell membrane. Then estrogen attaches to a specific receptor to form a complex that acts as a transcription factor by binding to a promoter region of DNA to enable RNA polymerase to bind for transcription to occur. This leads to the formation of proteins involved in cell division. Moving on. In part D, the expression of genes in cancer cells can be regulated by histone modification. So DNA that is not being transcribed is tightly wrapped around histone proteins. The diagram shows DNA wrapped around histone proteins. So we see there is somewhere where it's tightly wrapped, and we see somewhere where it's loosely wrapped. Those that are loosely wrapped could be transcribed if they're exposed more. Acetylation of histones is one example of histone modification. Acetyl groups are added to histone proteins. Suggest how acetylation of histone proteins could result in increased gene expression. Use the information in the diagram to support your answer. Remember acetyl groups linked to lysine. So I said acetylation leads to change in ionic charge of lysine. The charge is going to change from positive. This leads to a structure change. So the DNA is going to be loosely wrapped around the histones so that transcription can occur. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Aerobic respiration is a metabolic process involving a series of chemical reactions. How is water formed during aerobic respiration? Water is formed when oxygen receives a hydrogen ion. So that means the oxygen is being reduced. So water is formed during reduction of oxygen. In part B, the effect of exercise on the respiratory quotient of a cyclist was investigated. The cyclist undertook moderate exercise for 90 minutes. The graph shows the respiratory quotient for the cyclist during this investigation. So we have respiratory quotient on the vertical axis and time in minutes on the horizontal axis. And we can see as time increases, there is a decrease in the respiratory quotient. Moving on. Here they say, the rate of oxygen uptake by the cyclist at 60 minutes is 2.20 decimeters cubed per minute, which is the rate of carbon dioxide production by the cyclist at 60 minutes. So we go back to the graph. At 60 minutes, we can see the respiratory quotient is 0.88, as stated here. So I'm going to use that. But we know that respiratory quotient is carbon dioxide released divided by oxygen used. So since RQ at 60 minutes is 0.88, we can say 0.88 is equal to the carbon dioxide released divided by the oxygen produced, which is that. So making this the subject, my answer was 1.936, which is approximately 1.94 decimeters cubed per minute, and therefore B is my answer. Next they say, the table gives a respiratory quotient for aerobic respiration using fats or carbohydrates as respiratory substrates. So the substrate we have here is fat, and the respiratory quotient is 0.7. For carbohydrate, the respiratory quotient is 1.0. They say, explain the changes in respiratory quotient in this person during the exercise period of this investigation. They want you to use the information in the table and the graph to support your answer. Going back to the graph, we can see as time increases, the respiratory quotient decreases. So a decrease in respiratory quotient means we are metabolizing more fat because it has a lower respiratory quotient than carbohydrate. So my second answer was the decrease shows that more fat was used for respiration than carbohydrate. Moving on. In part C, chronic obstructive pulmonary disease is a life-threatening illness. This disease greatly reduces the ability to take in oxygen in the lungs and is a risk factor for lung cancer. The graph compares the incidence of lung cancer in smoking and non-smoking individuals with and without COPD. So on the vertical axis, we see the incidence of lung cancer in percentages, and down here we see age in years. So we can see, as age increases, individuals with COPD have a high incidence of lung cancer in comparison to individuals without COPD. And when we compare smokers and non-smokers, we can see for those without COPD, smokers have a high incidence, and those with COPD, smokers have a high incidence. So if you're a smoker, there is a higher chance of getting lung cancer. In South Korea, there are 105,500 males and 158,600 females in the 80-year-old age group. Calculate the total number of people in this age group that will have lung cancer who are non-smokers with COPD. So I came to the graph at 80, non-smokers with COPD, which corresponds to that, and my percentage was 7.6%. They want us to give our answer in standard form. So total number of people in the 80 age group are going to be that plus that, giving me 264,100. And since my percentage is 7.6%, so 7.6 divided by 100 times that gave me 20,071.6, which corresponds to 2.0 times 10 to the power 4 in standard form. So my answer is that, although the allowed range is between that and that. Moving on. Here they say, cigarette smoking causes inflammation in the lungs. This can result in damage to the walls of the alveoli and lead to COPD. Cigarette smoking also causes the production of thick mucus in the lungs. Deduce the effect on the heart rate and ventilation rate in a person who smokes and has COPD. Now we have seen it causes thick mucus and then it damages the walls of the alveoli, meaning we're going to have less surface area for gaseous exchange. So I say damage to the alveoli will reduce the surface area for gaseous exchange, and production of thick mucus will block the alveoli and increase the diffusion distance so less oxygen will reach the tissues. So the heart rate and ventilation rate will increase so that sufficient oxygen and nutrients are delivered to the blood. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Skeletal muscle contains slow-twitch and fast-twitch muscle fibers. The table compares the percentage of slow-twitch fibers in different types of athlete. So the type of athlete, we have a 100-meter runner. These are sprinters. The range of percentage of slow-twitch fibers in their muscle is 12 to 30%, meaning the other percentage, which could be about 70%, is going to be fast-twitch. Then 1,500-meter runner, they have 30 to 65. 10,000-meter runner, 60 to 95. And the marathon runners are going to be 60 to 95 as well. The key thing is, as distance increases, the percentage of slow-twitch muscle fibers increases. So they say explain the importance of slow-twitch muscle fibers in these types of athlete. We know slow-twitch muscle fibers have more myoglobin and they have more mitochondria. Myoglobin is going to provide more oxygen. The mitochondria are going to provide more energy in order for aerobic respiration to occur. So I say slow-twitch muscle fibers are vital for long-distance runners. We can see that from the table. The longer the distance run, the higher the percentage of slot-rich fibers. Slot-rich muscle fibers contain numerous mitochondria, more myoglobin, and numerous capillary networks, and these allow for aerobic respiration for ATP synthesis. Moving on. In part B, name the basic unit of contraction in a skeletal muscle. The basic unit of contraction is a sarcomere. In part C, the heart pumps oxygen-rich blood to the tissues. Cardiac output and cardiac index can show how effectively the heart is working. A patient had a heart rate of 65 beats per minute and a stroke volume of 57 centimeters cubed per beat. Calculate the cardiac output for this individual. They want you to use the equation stroke volume is equal to cardiac output divided by heart rate and give your answer in decimeters cubed per minute. So using this formula, my cardiac output is making this the subject is going to be that times that. So it's equal to stroke volume times heart rate. My stroke volume is that, which is 57 times the heart rate, which is 65, giving me 3,705 centimeters cubed per minute. But my answer should be in decimeters cubed per minute. To convert centimeters cubed to decimeters cubed, we divide by 1,000. So dividing this by 1,000, I get 3.705. You could write this at 3.71 or 3.70, or you could also write it at 3.7 decimeters cubed per minute. So whichever you choose, I could have written a 1. Actually, let me write a 1 here to make it better to round off. 3.71. I'm going to make this a 1 as well. 3.71. Rounding off, when I round off this to that, to be 1. But you could also have written 3.705, or you could have written 3.7 decimeters cubed per minute. Moving on. Here they say the cardiac index can be calculated using the formula, which we have here. The table shows the data for 2 patients. So we have patient A and B, the heart rate for each, the stroke volume for each, and the body surface area. So they say the cardiac index for patient A was 2.92 decimeters cubed per minute per meter squared, which is the correct cardiac index for patient B. I'm going to substitute in this formula. So cardiac index should be the stroke volume for patient B, which is that, times the heart rate, which is that, divided by the body surface area, which is that. So my answer was 2.47907995. Rounding it off to 3 significant figures like they did here, I got 2.48 decimeters cubed per minute per meter squared, and B is my answer. Moving on. Here they say state the ratio of the body surface area of patient A to patient B. If you go back to surface area, patient A and B, we have the ratio which is that to that. So 1.54 to 2.39. The ratio becomes 1 to 1.55, and that is my answer. In part D, the cardiac output changes in an athlete as their running speed increases. The graph shows cardiac output for an athlete running at different speeds. So on the vertical axis, we have cardiac output in decimeters cubed per minute. In the horizontal axis, we have running speed in miles per hour. Overall, we see as running speed increases, the cardiac output increases. So next they say, comment on the effect of running speed on cardiac output. So we have seen that as running speed increases, cardiac output increases. And also we saw a maximum output is obtained between 10 to 12 miles per hour. If we can see that, between this range, that is where we obtain the maximum. Based on the results, we could say this is the maximum. But if data was collected at more smaller intervals, maybe the maximum could be here or it could be here. So to be general, I just say it between that and that. Because we see still increasing and then here it levels off. So we can see it's between that and that. So next they say, exercise increases the rate of respiration in muscles. Explain how an increase in the rate of respiration causes an increase in the heart rate of an athlete. When the rate of respiration increases, more CO2 is produced, and that causes the decrease in the pH of blood. So receptors are going to detect that and send impulses to the brain, and then a response is going to be carried out. So I said increase in the rate of respiration increases CO2 concentration in blood, so the blood pH decreases. Then the chemoreceptors detect this pH decrease, and they send impulses through the sensory neurons to the cardiovascular control center in the medulla. A response stimulates the SAN for faster depolarization, and this causes an increase in the heart rate. Moving on. Here they say anaerobic respiration produces lactate. Describe what happens to this lactate. The lactate produced has to be taken to the liver where it's going to be converted to pyruvate, and then it enters the Krebs cycle through the link reactions. So I said lactate is transported to the liver. In the liver, it is converted to pyruvate. In the presence of oxygen, pyruvate enters the link reactions for aerobic respiration. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Scientists investigated the effect of sucrose solutions on the desire of rats to eat. The release of dopamine in some areas of the brain increases the desire of the rat to eat. Scientists measured increases in the release of dopamine in the brain of rats given different concentrations of sucrose solutions to drink. So this is the data given. We can see dopamine percentage increase on the vertical axis and then the concentration of sucrose solution on the horizontal axis. Overall, we see as the concentration of sucrose solution increases, we see an increase in the percentage of dopamine that is produced. We see that error bars were put, so the experiments were repeated. However, there is overlap, meaning there is no significant difference in these results at the different concentrations since overlap exists. So here they say, the scientists concluded that drinking a solution of sucrose had an effect on the desire of the rats to eat. They say comment on the results shown in the bar chart and how they support this conclusion. So again, now to just read what I've explained. The higher the concentration of sucrose solution, the higher the percentage of dopamine released, and this leads to rats having more desire to eat. However, error bars at different sucrose concentrations overlap, so there is no significant difference in the percentage of dopamine produced at different sucrose concentrations. Moving on. Here they say the table gives the results for a sucrose concentration of 0.4 mol per decimeter cubed. They say complete the table to calculate the standard deviation for these results. They want you to give your answer to two significant figures. From the results, we see there is x, but there is x minus x bar, which is negative 15. If this is 45 and x minus x bar is negative 15, it means x bar is going to be 60. So 45 minus 60 gives us negative 15. And therefore, we can be able to find this result here. It should be 58 minus 60, giving us negative 2. And when we square this, we get a 4. This value here is the sum of all this. So add that plus that plus that plus that. I got 434. The next one is n minus 1. We see the number of results are 1, 2, 3, 4. So they should be 4 minus 1, giving us 3. And then s, which is from here, is about dividing that by that and getting a square root. But since they say to two significant figures, I rounded it off to 12. Moving on. In part B, when humans are in danger or under stress, they respond by activating the fight-or-flight response. Describe the role of adrenaline in the fight-or-flight response. We know adrenaline is going to be produced from the adrenal glands and is released into the blood. It will bind to receptors on the surfaces of cells of target organs. Here it stimulates physiological changes in the organs like increasing the heart rate or conversion of glycogen to glucose in the liver. It could cause vasodilation in the skin, and this is to release excess heat during exercise. So any two given physiological changes were awarded 2 marks. Moving on. In part C, negative feedback is involved in maintaining systems within narrow limits. The blood glucose concentration has to be maintained within narrow limits. Insulin and glucagon are hormones involved in the control of blood glucose levels. The diagram shows how blood glucose concentrations are controlled in the body. Also, we see a pancreas that produces 2 hormones, either glucagon or insulin. When glucagon is produced, it can cause the conversion of glycogen into glucose, and when insulin is released, it can cause the conversion of glucose into glycogen. When glucose is converted to glycogen, the blood glucose decreases, and when glycogen is converted to glucose, the blood sugar raises. So down here they say the graph shows the daily changes in the concentration of insulin, glucagon, and glucose. On the vertical axis, we have blood glucagon concentration, blood glucose concentration, and blood insulin concentration after a meal on the horizontal at specific times. So here they say discuss how negative feedback is involved in the control of blood glucose concentrations. They say use the information in the diagram and the graphs to support your answer. So here I began with a diagram. From the diagram, the pancreatic cells secrete hormones, insulin and glucagon, and both hormones work on the liver cells. From the graph, the concentration of blood glucose increases after a meal. Also, insulin concentration increases after meal. Blood glucose concentration has to be controlled within a narrow range for proper function of the body. Changes in blood glucose concentration are detected by receptors on the cells of the pancreas. So if high concentration of glucose is detected, insulin is produced. Glucagon is produced if the blood glucose concentration is low, so that glycogen is converted to glucose. Both insulin and glucagon act as transcription factors. Insulin stimulates the liver and muscle cells to convert glucose to glycogen for storage, while glucagon stimulates the breakdown of glycogen into glucose. Once the blood glucose concentration returns to normal, the concentration of insulin or glucagon will be decreased. So this brings us to the end of question 7. Let's continue to question 8. To answer question 8, we'll begin with a scientific article. I'm going to scan through the article as you visualize, and then I'll come to answer the question. So this is the article for answering question 8. It goes like this. It's about brain imaging, neurodegeneration, and mitochondria. We can see talking about free radicals and how they cause aging. Mainly reactive oxygen species from the mitochondria. As you're going through this, you have to pause so that you can read slowly. Now, question 8 says, the scientific document you have studied is adapted from the article Brain Aging and Neurodegeneration from a Mitochondrial Point of View by Amandine Grimm and Annette Eckhart. Use the information from the article and your own knowledge to answer the following questions. Question A says, suggest why there is a relationship between the aging process and neurodegeneration. This is from paragraph 1 and 2. Reactive oxygen species are produced in the mitochondria during aerobic respiration. So these cause damage to more mitochondria, and damaged mitochondria are removed by autophagy. This will lead to fewer mitochondria. So less energy is going to be produced, leading to fewer impulses moving across the synapses or leading to damage to nerve cells. So neurons will be cleared. This alters the brain chemistry, like decreasing the production of neurotransmitters, which include dopamine as well as serotonin, resulting in neurodegeneration. Moving on. In part B, describe how energy released from glucose is used in oxidative phosphorylation. This is from paragraph 2. So we know that there are coenzymes that are reduced, like reduced NAD and reduced FAD. These will transport hydrogen ions and electrons to the electron transport chain. So the electrons transition through the ETC, which is the electron transport chain. The energy released is used to pump the protons into the intermembrane space. This is from the matrix. Electrochemical and potential gradients are created, and the protons return into the matrix through ATP synthase. Here, ADP and PI combine to produce ATP, which is phosphorylation. In part C, explain how free radicals, which are the reactive oxygen species, can cause damage to mitochondrial DNA. This is paragraph 2 and 4. Reactive oxygen species are produced in the mitochondria during oxidative phosphorylation. These could cause mutation to mitochondrial DNA. We also need to know these breaks or mutations are caused because hydrogen and phosphodiester bonds may fail to form at certain points. So repair proteins can't repair these faults in the mitochondrial DNA, so it is going to be damaged. Moving on. In part D, the brain is a remarkable organ composed by highly differentiated cells with different morphology according to their role and their localization. Describe how this differentiation can occur. This begins with stem cells. These are brain stem cells that are going to be differentiated into specialized cells that perform specific functions. This is going to be achieved by some transcription factors like chemicals or hormones that cause gene expression for transcription of specific genes into messenger RNA and translation of messenger RNA into proteins. The produced proteins will determine the structure, the function, and location of some brain cells in a specific region of the brain. In part E, explain what is meant by the phrase mitochondrial proteome. Proteome is about proteins, so these are proteins either found in the mitochondria or produced in the mitochondria or functioning in the mitochondria. And in part F, explain why synaptic mitochondria are more susceptible to aging than mitochondria found in other parts of a neuron. The synapse is very active and requires a lot of ATP generation for active transport, so the mitochondria in the synapse are working so hard. So I say synaptic mitochondria have higher rates of oxidative phosphorylation due to the great ATP requirements for active transport. So more reactive oxygen species are produced, and this leads to greater damage and faster aging. In part G, explain why the mitochondria genome is essential for oxidative phosphorylation. This is paragraph 10. The mitochondria genome contains genes that are necessary for formation of specific proteins that are going to be involved in phosphorylation. These proteins could even include ATP synthase. So I said mitochondrial DNA contain genes needed for mitochondrial function, like synthesis of proteins involved in the electron transport chain, like protein pumps, and proteins involved in oxidative phosphorylation, like ATP synthase, and the combination of ADP and PI to form ATP. And lastly, suggest how damaged mitochondria are removed by autophagy. In paragraph 11, for autophagy to occur, a vesicle is going to be formed around the mitochondrion, and then this vesicle is going to fuse with a lysosome containing lytic enzymes. Then lytic enzymes are going to break down the mitochondrion. So I said by formation of a vesicle around the damaged mitochondrion, followed by fusion with a lysosome containing lytic enzymes. This results in digestion of the damaged mitochondrion. So this brings us to the end of question 8, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.